Okay, Mitch, do you want to go ahead and share your computer? And we'll get started and people will come in. Want to welcome everybody to this month's seminar series for the CIHD. And I'm going to just be very brief here and tell you we are so happy and pleased to have Mitch Knutson speaking to us. Mitch is currently a professor of nutritional biochemistry and food sciences and human nutrition at Gainesville, Florida, University of Florida there. And Mitch, what I remember distinctly about Mitch, and I want to just say this one thing and nothing else, is I was at, I believe, maybe at a Gordon conference where I remember a iron scientist at the meeting turning to me and say, Mitch has got the transporter that we've all been looking for, this Zip 14. And so that was a big hit on the horizon of iron research. And I'm so glad that you are able to present to us today. I want to invite everyone to mute, populate questions in the chat, and we will, I will monitor those questions. If they seem appropriate for sort of a timely interruption, I will do so. Otherwise, we'll hold questions to the end. And please feel free to write those in there and I will kind of survey them for us. And again, Mitch, thank you so much for talking today.
All right, thank you, Diane. It is a pleasure to be invited here. And I have tailored my talk to be pretty much on time, 45 minutes. So we will have time for questions at the end. But if there's a burning issue, please feel free to interrupt me. Okay, let me just see if I'm, so for some reason, okay, all right. So today I'm going to talk about Zip 14 as a potential therapeutic target in the treatment of iron overload. And most of the stuff I'm going to show you today is unpublished. Of course, I'm going to have a little bit of background, very limited amount. But just to make sure we're all on the same page. So first, about iron overload. It's commonly divided into either primary iron overload, which is known as hemochromatosis. This is the inherited form. And there's also secondary iron overload, which is not inherited. So when we talk about primary iron overload, we're talking about mutations in genes. Most hemochromatosis, as you know, is resulting from the C282Y mutation in the HFE gene, greater than 85% of cases. But then again, we have mutations in other genes, such as hemojuvelin, hepcidin, transferrin receptor 2, ferroportin. These all result in various forms of hemochromatosis. Mutations in these two genes, hemojuvelin or hepcidin, result in a particularly severe form of hemochromatosis, juvenile hemochromatosis. And one of the things that I would like to point out, because it's relevant to the talk, and it's kind of just a housekeeping nomenclature thing, is that many of you are probably aware that the official gene symbol for hemojuvelin changed a few years ago from HFE2 to the more apt hemojuvelin. Nonetheless, some of my data that I'm going to show that's been published was done using the HFE2 designation. So just make a mental note of that, and I'll come back to that later, because we use the hemojuvelin knockout mouse as a model in a lot of our studies. When we talk about secondary iron overload, one good example is thalassemia major, hemoglobin disorder resulting in abnormal hemoglobin production. And this, like thalassemia, beta thalassemia, for example, is usually diagnosed in infancy. And to survive, these infants require regular and frequent repeated blood transfusions, which over time invariably lead to iron overload and an early or earlier death. So let me just kind of do the bird's eye view of hemochromatosis and just talk about what's going on with the main players involved in iron metabolism. We have the bone marrow, the liver, the macrophages, particularly macrophages of the the liver, spleen, and the bone marrow. These are the reticular epithelial macrophages, which eat old red blood cells. And then the iron is basically recycled back to the bone marrow for new red cell synthesis. So we absorb iron from our diet, and normally we can keep this in balance by just regulating how much iron we absorb. But in hemochromatosis, we don't do that. We tend to hyperabsorb dietary iron, and this is usually the result of the inability to produce the iron regulatory hormone hepcidin. So hepcidin, as you all know, blocks iron uptake from the duodenum. It inhibits iron release from macrophages. But in hemochromatosis, if you don't have hepcidin or make too little of it, what happens is you hyperabsorb dietary iron. Now, this is animated here, so you can follow along, is that in hemochromatosis, you hyperabsorb dietary iron, and you also release more iron from reticular endothelial macrophages. Both these things increase the amount of iron that's carried on plasma, iron transport protein transferrin, or TF, oftentimes exceeding its carrying capacity, which leads to the presence of labile plasma iron, also known as non-transferrin bound iron. And that non-transferrin bound iron, or NTBI, is taken up mainly and rapidly by the liver, where it contributes to iron loading of this organ. And it's also could be taken up by, for example, the pancreas and the heart. And not surprisingly, in hemochromatosis, these are often the first tissues that are affected with iron overload related pathology. What my lab has been interested in the past 15 years or so is to try to figure out what are the transporters that are involved in NTBI uptake into various tissues. And the transporter that we spent most of the time on is ZIP14. The official gene name is SLC 39A14. Throughout this talk, you will see me use either the term ZIP14 or the official gene name, but from here on out, I'm just going to refer to it as ZIP14 to save 5 syllables each time. So ZIP14. ZIP14 is a member of the ZIP family of metal ion transporters. They're called metal ion transporters because they transport a variety of divalent cations. In mammals, they were first identified as zinc transporters, and they're often associated with zinc transport, but we showed in 2006 that ZIP14 could transport not only zinc, but it could transport iron as well. And later studies in collaboration with Brian McKenzie's group, we showed that it could transport zinc, iron, manganese, cobalt, and also cadmium, and likely some others. An important feature of ZIP14, which differentiates it from the other well-known iron transporter, DMT1, is that ZIP14 transports iron optimally at pH 7.4, which makes it a very good candidate for transporting NTBI out of the plasma and into tissues. DMT1, as you know, transports iron optimally at acidic pH and is probably not very efficient at transporting it at pH 7.4. Hence, ZIP14 actually is the main NTBI transport protein in vivo, at least for some tissues. And this we showed back about 6 years ago by using the ZIP14 knockout mice. More specifically, we used radio tracer of iron 59 to basically see where the iron went, and it wasn't taken up into the liver and the pancreas. And so definitely ZIP14 plays a role in NTBI uptake in vivo. But more importantly, I guess, physiologically speaking, this study, we also found that ZIP14 is absolutely required for iron loading of specific organs and cell types. And how we show that was a variety of ways. We used different models of iron overload. We used HFE knockout mice. We used hemojuvelin knockout mice, we used dietary iron overload. And I'm just going to show you, I think, 3 slides of data from that paper because it sets the stage for and rationale for why we're doing what we're doing now, looking at it as a therapeutic target. And so one way we looked at its role in iron accumulation is we basically crossed ZIP14 knockout mice with hemojuvelin knockout mice to generate the double knockout mice. So we know that hemojuvelin knockout mice will hyperabsorb dietary iron and load significant amounts of iron even by 4 weeks of age, 6 weeks of age. The question we had is, if you have an hemojuvelin knockout mouse and you also don't have ZIP14, how does it load iron? Where does it load iron and how much? And so here are some of those data. If we look at tissue non-heme iron levels, and here are the liver non-heme iron values, you can see that the hemojuvelin knockout mice loaded, this is at 6 weeks of age, loaded about 10 times normal amounts of non-heme iron in the liver. In sharp contrast, the double knockout mice lacking ZIP14 loaded much less, very similar to control mice. In other tissues, such as the pancreas, we saw a similar thing. Hemojuvelin knockout mice accumulating iron in the pancreas, but the double knockout less. The story got even more interesting when we looked at other tissues, such as the heart, the kidney, and the spleen. What we saw was that iron was actually increased in these tissues, most notably the kidney, if you look at the amount wise, and also the spleen, and less so the heart, but still in the heart. So it looks like from these data that basically in the double knockout mice, well, if you just take the single knockout mouse, the hemojuvelin knockout mice, normally the iron will load first in the liver and the pancreas and less so in the heart. But if you can't load iron in the liver and the pancreas, such as in the double knockouts, or load less, where does it go? Well, it goes to other organs, such as the kidney, spleen, and the heart. We did the same type of experiment, just looking at dietary iron overload. I'm in a nutrition department. We like to feed mice different diets. So we fed mice starting at weaning age, an iron overload diet, a 1% carbonyl iron diet for 4 weeks. And what we see again, we see iron loaded mice or iron overload, FEO diet, loading a tremendous amount of iron in the liver, less so if the mice lacked ZIP14. In the pancreas, they did load some iron in the pancreas, but more so in the kidney and the spleen. If you look at the amounts, most of the iron that didn't load in the liver loaded in the kidney, in the spleen, and the heart and the pancreas. So if we focus now just on the liver and ask the question, where in the liver does the iron load? I mean, you do see iron loading in the ZIP14 knockout mice. I told you that it's required for iron loading, right? Well, where it is loading, if you compare it to the control, but the interesting part is, where is it loading? And so if you look at Pearl's stain of these liver sections, we see that the wild type mice fed an iron loaded diet. They load iron in mostly in periportal hepatocytes, very nice vibrant blue staining. But in ZIP14 knockout mice, you see iron loading, but it's a totally different pattern. The most striking thing is you don't see iron loading in hepatocytes at all by Pearl's stain. Where you do see it is you see it loading in these non-parenchymal cells, some of which are Kupffer cells. We showed this in the paper. You'll probably know that Pearl stain is not very sensitive. So even if you use DAB enhanced pearls and enhance it even further using cobalt, and here are the data there, you still don't see iron loading in hepatocytes, which is quite remarkable, but you do see it loading in these non-parenchymal cells. We also looked at where iron is loading in the pancreas. I'm not going to show you those data, but bottom line is it's similar to the liver. It's loading a little bit in the pancreas, but not where it normally does. It normally loads in the acinar cells, but in the ZIP14 knockout, it doesn't. It loads in collagen for whatever reason, a small amount in collagen. That's where we detect it, very little, if any, in the acinar cells. So the conclusions from that paper that we published, these are the main two conclusions, is that what is ZIP14 doing in iron overload? Well, in the liver, it's taking up NTBI from the circulation. ZIP14 localizes to the sinusoidal or basolateral or blood-facing membrane of hepatocytes where it takes up NTBI. It also localizes to the plasma membrane of acinar cells where it takes up NTBI. So this is where we were in 2015. And so the next things that we did, among the things that we did, we decided to look at more tissue-specific knockout models of ZIP14. And the first tissue we looked at was the liver, because that's where ZIP14 is most abundantly expressed, in part to confirm what we saw with the whole body knockout, and also to see if we could see anything interesting or new or novel. And so our next study, we did classical hepatocyte-specific knockouts using the Albie-McCree transgenic mice. And so we took the flox mouse, crossed it, and we have the Cre-negative mice, which are used as controls, and the Cre-positive, which are the mice that are deficient in ZIP14, specifically in hepatocytes. And what we did is we did a simple, quick dietary iron overload study where we basically took the control mice plus the hepatocyte-specific knockout, fed them either an iron control diet, a purified diet, 50 ppm iron, or an iron overload diet, 1% carbonyl iron for 4 weeks. That's our typical format. Our group designations here, FEC is iron control, FEO is iron overload. We use 6, that should be 2 to 10 mice per sex per genotype, and they were litter mates. And here are the plasma iron indices. I'm not going to go through all of these. I think the most, you see, the most relevant data for the talk is right here, the transferrin saturation. Bottom line is that the mice fed the iron overload diet, their transferrin saturations were at or above 100%, indicating that these mice most probably have a lot of the NTBI in the plasma. So what happens to liver non-heme iron levels? Well, as you could probably imagine or predict, is that the hepatocyte-specific knockout mice fed an iron overload diet accumulated much less iron in the liver when compared to control mice. What happens if we look at pearl stain? And here is the control mouse fed an iron overload diet, the pearls, and here's cobalt dab enhanced pearls. We see, as expected, most of the iron is loading in periportal hepatocytes, less so towards hepatocytes in the central lobular region. See a much different pattern in the hepatocyte-specific knockout mice fed an iron overload diet. I mean, you can't really detect any iron by pearl stain, cobalt dab enhanced. You could start to see some faint staining, what looked like hepatocytes in the central lobular region, but most of the iron was found in these non-parenchymal cells, most likely Kupffer cells. So bottom line is that the distribution of iron in the total body knockout is very similar to what we see with the hepatocyte-specific knockout. So it's clear that I think we've confirmed that ZIP14 functions most definitely in hepatocytes in the liver. What happens to tissue non-heme iron levels in other tissues? I mean, I know this is a really busy slide, lots of data, but I showed you these liver iron levels already. This is the new information here. And I think it's pretty clear, you can see what's going on, is that in these mice lacking ZIP14 and hepatocytes, when they're fed an iron overload diet, the iron doesn't load in the liver, it loads in other organs. Mostly the pancreas, 16 times normal. Also the kidney, 11 times normal. The heart, 5 times normal. Brain and the spleen. So everywhere we look, it's higher, except for the liver. So you're thinking, well, this is, what's really new here. In my mind, what's new is it kind of shows you the hierarchy of iron loading. If you really can't load iron, where it normally loads first, and that's hepatocytes. Where does it load? Well, it loads in the pancreas and the kidney, and then the heart and other tissues.
Mitch, I'm going to interrupt
Yes.
for a question from Carolyn Philpott. Do you want to ask it, Carolyn?
Sure. Thank you. Can you hear me?
I can hear you.
So Mitch,
Yes.
you showed lovely pictures of the DAB stain of these livers with iron overload when you have ZIP14 knocked out in hepatocytes. Are they accumulating iron in the sinusoidal endothelial cells?
Yeah, that's a great question. My answer would be yes. And the reason why it's yes is because they have an increase in hepcidin production. Hepcidin production is the same as in a wild type animal. So they probably, since endothelial cells make BMP6, right? And we.
I'm just looking at the pattern of staining.
Oh yeah, well, we didn't use the markers.
I'm just wondering, what are we looking at? Yeah.
Well, just based on the mRNA levels of BMP6 and then hepcidin, I conclude that they're loaded in endothelial cells. Did we use an endothelial cell marker to localize it? No, we didn't. But yeah, that's a great question. It's probably loading there.
Yeah, I'm just trying to understand whether endothelial cells take up iron and hang on to it, or whether they take up iron and spit it back out.
Yeah. That's a great question. Something we kind of talk about frequently, about how endothelial cells take up iron and what do they do with it. So yeah, great question. Thank you.
Thank you.
Cool. Go ahead. Keep going. Thanks.
Going back to this, the thing that really jumped out at us when we looked at this was the heart data. And why it was surprising to us is when I saw that they loaded about 200 microgram per gram, I thought, wow, that's much different than what we saw with the total body knockout. They didn't load nearly as much as that. And let me show you what I mean, just kind of as an example. Just compare the heart non-heme iron concentrations in the liver specific versus the total knockout, which we published. So the liver specific, will have ZIP14 in the heart because it's knocked out only in the liver, whereas the total knockout will not have it in the liver or the heart. And if you just compare these two, you can see that when there is ZIP14, they're loading more iron in the heart. And you might be saying, well, maybe it's because not only because it's a different study, but maybe you have different iron loading in the liver or the whole body. So let me just show you the liver non-heme iron levels. They're very similar between these studies. And remember that these two studies were the identical design. It started at 4 weeks of age, fed them a 1% carbonyl iron diet for 4 weeks. And so it suggested to us that ZIP14 might be playing a role in iron accumulation in the heart. And so really the best way to test that is to make a cardiomyocyte specific knockout mouse and do the study the right way. And we did do that study, but I'm not going to show you those data because if you want to see those data, you're going to have to come to the ASH meeting in December. So we submitted an abstract to that, and I really can't tell you anything about it now. but please come to the meeting. This leads me to kind of the focus of the talk today, and this was reviewed by Stefano Ravella and colleagues, and he wrote about, kind of drug discovery and targeting proteins involved in iron metabolism. He wrote the central role of ZIP14 in transporting NTBI into tissues that are typically affected by iron overload, especially the liver and the pancreas, makes the inhibition of ZIP14 an appealing strategy for the clinical management of iron overload. Well, is it? Well, what is the current strategy for clinically managing iron overload? Well, it depends on which type of iron overload you have. We know that in hereditary hemochromatosis, it's phlebotomy. Juvenile hemochromatosis, it's also phlebotomy, and in severe cases, it's iron plus iron chelation. Whereas in thalassemia major, the treatment is iron chelation, no phlebotomy usually. And you can either use a variety of different iron chelators. Nonetheless, in thalassemia major, despite iron chelation, iron does still accumulate over time in tissues such as the heart. And so there have been additional strategies to kind of mitigate iron loading in the heart by using adjunct or add-on therapies. And one of those add-on therapies is by using an LTCC blocker, the L-type calcium channel blocker, amlodipine. It's widely thought that cardiomyocytes take up NTBI via L-type calcium channels. And so the idea is why not just try to block it by using these calcium channel blockers and NTBI uptake blockers, so to speak, plus iron chelation. This should be better. And there's a fair amount of data in animal models and even clinical trials that support the use of LTCC blockers to mitigate iron loading. And that made us think, well, if you can use LTCC blockers, why not use iron chelation plus a quote unquote ZIP14 blocker? And the rationale for such an add-on therapy would be that, okay, the liver and the pancreas are well known to take up NTBI and load iron via ZIP14. Let's say that we could block ZIP14. What would happen is that you would have an increase in plasma NTBI. And then if this was added in addition to an iron chelator such as deferriprone would bind to that iron and cause it to be excreted, and you would have a decrease in body iron, which is what you want with iron chelation. So the idea is that this would enhance maybe or accelerate body iron losses if we could just block it in these tissues. So one way that we thought of blocking, so to speak, ZIP14 function was to use antisense oligonucleotides, which are basically small stretches of 14 to 20 nucleotides that target complementary RNA. And so ASOs, for example, that target ZIP14 will bind specifically to ZIP14 mRNA, cause its degradation, leading to decreased ZIP14 protein levels. And so what we did is we investigated two different ZIP14 ASOs. We used ASO1 and ASO2. These were designed by and provided by Ionis Pharmaceuticals. And so our goal was to assess the efficacy of reducing ZIP14 expression in vivo as an adjunct or add-on treatment for iron overload. And the way we did this is we took hemojuvelin knockout mice and we gave them the standard treatment of iron chelation, and the iron chelator we used was deferriprone. The reason why is because deferriprone is an orally active one, and it's shown some promise at reducing cardiac iron levels. And as for an add-on treatment, we looked at ZIP14 ASO1 or ASO2. And so our hypothesis that we're testing is that ZIP14 ASOs plus DFP treatment, so the combined treatment would be more effective than DFP alone at reducing tissue iron levels or iron loading in hemojuvelin knockout mice. And the way we did that is we took hemojuvelin knockout mice. In this study, we only used male mice. We started at 7 weeks of age. We had 4 to 5 mice per group, and we had 7 groups here. This was our basically no treatment control. This was our scrambled ASO control. Then mice were given DFP alone, ASO1 or ASO2 alone, and then the combined treatments. They were given the treatments for 6 weeks. The DFP was administered or provided in the drinking water at this concentration. This was recommended by folks at Apel Pharma, and we also confirmed that this was optimal for hemojuvelin knockout mice. ASOs were given subcutaneously twice per week per Ionis instructions and recommendations. We also validated that this dose was very effective, at least in some tissues. And the data that I'm going to show you today is just the ZIP14 mRNA levels and the tissue non-heme iron levels. So here's the mRNA levels, and lots of groups here, lots of data, but if you could just focus on the ASO groups here, it's quite clear that if you look at ZIP14 mRNA levels, the ASOs are working. They're decreasing and pretty dramatically the expression of the mRNA, 94% with ASO1, less so with ASO2, and here's the combined. We also got good knockdown efficiency with kidney, ASO1 about 84%, ASO2 about 80%. And then we have what we call our more second tier tissues in terms of efficacy, and that would be the spleen and the heart, where we got about 70, 60, 50%, depending on the ASO. So, and then the pancreas, we didn't find any a decrease in expression, nor really in the brain. So overall, they were effective at decreasing ZIP14 in the liver and the kidney, somewhat in the spleen and the heart, but not so in these tissues. What happens to mRNA or non-heme iron levels? And there's a lot of data here, a lot to process, but I'm just going to walk you through basically tissue by tissue. The liver here, if we just look at deferoprone treatment. Now, the thing to point out is, remember that these are all hemojuvelin knockout mice. And the dotted line here is the control value. I'm not showing you the control group, this dotted line. So a normal mouse has about 100 micrograms of non-heme iron per gram tissue. You can see that the hemojuvelin knockout mice are massively iron loaded. You give them deferoprone, it goes down by about 40%. If you just give them ASO1, non-heme iron levels go down by 93%. ASO2, less effective. ASOs plus DFP, even more effective than ASO1 or DFP alone. So what happens in other tissues? If we look at the pancreas, we see that deferoprone decreased iron concentration in the pancreas, but ASO1 increased it. So ASO1 decreased iron in the liver, but it increased it in the pancreas. ASO1 decreased in the liver, increased it in the pancreas, kind of like the knockout mouse. On the other hand, the combined treatment mitigated the iron loading in the pancreas. ASO1 and ASO2 both mitigated it there. If we look at the kidney, we see a very similar thing. DFP decreased it by about 50%, but ASO1 increased it. Combined treatment brought it back down to similar levels to DFP, and same here. I think you're starting to see the pattern. The heart, I think, is kind of interesting because you didn't see really much of an increase with the ASO treatment alone. In the heart, deferoprone treatment alone caused it to decrease about 20%, although this is not statistically significant when compared to the PBS or the ASO control. What's interesting is that the combined treatment with ASO1 or ASO2 decreased it significantly, at least compared to the PBS. And the spleen, similar things going on there. I'm not going to really go into that too much. So that's the data from tissue iron levels. So another thing we looked at, I mean, if you go back and look at this and think about it, and this is actually a question that I got several years ago from Ella after I presented some data on, at the East to West Iron Club. She asked me if I, so you see that iron is not loading as much in the liver with the ZIP14 knockdown here, but it's loading more in the pancreas. Well, are you really decreasing the total amount of iron, or are you just shuffling it around in vivo? That is this iron decreasing the liver but redistributed in the kidney and other organs? In effect, are you not decreasing total body iron? So what we did is we basically added up all the non-heme iron levels from the organs that we measured. And in this study, we took liver, pancreas, spleen, kidney, and heart. And we measured, or we calculated, the total non-heme iron. So we took the tissue concentration, multiplied it by the weight to get how much total non-heme iron was in there. And then we basically just added up all the tissues to get what we call total organ non-heme iron. That is the total organs that we measured. And here's what you see, is you see that in just an untreated mouse, they have about, in these organs, total about 2,400 micrograms of non-heme iron. Deferiprone cut that in about half. What's interesting is ASO1 by itself, no iron chelator, decreased it by about half, and the combined treatments even more so than DFP alone or the ASO treatment alone. So these data suggested to us, this kind of was surprising, that either the ASO1 treatment is causing an increase in total body iron loss, or maybe it's distributing to other organs that we're not measuring, or possibly even the carcass, such as muscle or other tissues. Unfortunately, we didn't measure that in this study, but this is what we find. So here's a kind of a recap of the main findings from this part, is that ZIP14 ASOs potently decrease ZIP14 mRNAs in the liver, decreasing hepatic iron concentrations by about 90%. That's good. On the other hand, ASO treatment causes iron to load in extrahepatic tissues, such as the pancreas, the kidney, and the spleen. This, we think, likely reflects a redistribution of iron from the liver. The addition of deferiprone to the ASO treatment mitigates iron loading in these extrahepatic tissues. The combined treatment decreased cardiac iron, whereas DFP alone did not. I think it's somewhat interesting. And the combined treatment was more effective than was DFP alone at reducing what we call total organ non-heme iron.
So Mitch, I'm
Yes.
interrupting again.
Yes.
So is the iron in the blood, does that go up? I mean, is the iron still circulating in extracellular tissues?
With the just ASO treatment alone?
Yeah.
Or
the ASO plus DFP?
Well, we didn't measure it. We didn't measure it. You know, we can't really measure non-heme iron because of the DFP in there, right? I guess we could measure it by ICP mass spec, but it's kind of a difficult thing to measure. So does that answer your question, Caroline?
So that's just
kind of.
So I'm just wondering, have you succeeded in getting a mouse that excretes iron, or not clear yet?
I'm getting to that.
Okay.
Yeah, getting to that.
Okay,
so this last point, ASO treatment alone was effective as DFP alone. So overall, from this study, it looked pretty promising. That seems like it has an effect, seems to enhance iron losses, or at least in some tissues compared to DFP alone, but there was an issue. There was a concern or a potential limitation. And so the potential limitation, just like to say that although ASOs can reduce ZIP14 levels, which decrease tissue NTBI uptake, at least some tissues, the thing to keep in mind is that NTBI is not physiologic. It's really a pathologic species. So that the concern is that if you reduce ZIP14 levels, maybe you're adversely affecting the physiologic function of ZIP14. So the question is, what is the physiologic function of ZIP14? And probably the best characterized physiologic function is its role in manganese metabolism. So manganese is similar to an NTBI in that it's transported by ZIP14. And so under physiologic circumstances, that's probably its most likely main substrate, manganese, because you don't have NTBI normally. Manganese is taken up in hepatocytes. Excess is secreted into the bile and excreted in the feces. Another way to look at this is that if you kind of look at the intestinal axis, so manganese is absorbed into the portal circulation, taken up by ZIP14, and is excreted in the bile when it's in excess. If you don't have ZIP14, what happens in terms of manganese metabolism is that the manganese bypasses the liver, enters the systemic circulation, where it can then deposit in extrahepatic tissues. And we showed this in our paper in 2018, where manganese homeostasis is indeed dramatically altered in ZIP14 knockout mice. This is showing manganese concentrations in the liver. They're down, which makes sense because that's where ZIP14 is functioning with manganese in the liver. It takes it up. So you don't take it up in the liver. What happens? It loads in extrahepatic tissues, most notably the bone, the brain, the kidney, and then other tissues to a lesser extent. They load in these tissues because whole blood manganese levels are elevated. So that's why, and this is what you see. When you have high blood manganese levels, it oftentimes loads in the brain, and this is bad because too much manganese in the brain leads to a Parkinsonian disorder known as manganism. So manganese loading in the brain is not a good thing. So what about our mice, the ASO treated mice? Are they having any effects on manganese levels in the liver or the brain? Well, here's the liver data. You can see that you knock down ZIP14, you decrease manganese in the liver. That's what you would expect. What happens in the brain? Well, you accumulate manganese in the brain, which is kind of what you would expect. What's kind of surprising is that the ASO1 plus DFP loaded more manganese in the brain than the ASO control, and saw the same thing here with this other DFP. You may be thinking, oh, these increases are relatively modest, they shouldn't have much an effect, but people in the manganese world make a case that even small increases in brain manganese, like twofold, are associated with changes in brain function and toxicity. Clearly, ASO1 plus DFP is not a good thing. You're increasing brain concentrations of manganese sixfold at least. So this is a concern. And so the question we had at this point is it possible to reduce ZIP14 expression in such a way that you can still enhance DFP-mediated removal of iron, but not affect manganese homeostasis?
Mitch, one question here,
Yes.
Before you get to that. How do you know that NTBI uptake is actually not involved in iron uptake into hepatocytes in the portal circulation? So did you try to measure whether you alter iron absorption into hepatocytes along the portal circulation?
That was in our cell metabolism paper. We measured NTBI uptake after oral gastric gavage, and iron 59, it was deposition in the liver was markedly less in the ZIP14 knockout mouse and markedly more in extrahepatic tissues. So it entered via the portal circulation, bypassed primarily the liver, got taken up by other tissues. So it's NTBI.
No, what I mean is even in normal mice, not in
normal mice,
normal
I think the iron goes from the portal blood to the liver mainly via transferrin. I think Paul Lindahl's data pretty showed that.
So there is no involvement in normal absorption by in ZIP14.
It may be in early life, because we do see a decrease in hepatic iron concentrations in ZIP14 knockout mice at 4 weeks of age compared to wild type mice. But at later ages, that difference disappears. So maybe in early life, there's NTBI.
Okay, thank you.
Thank you. So can we optimize this situation? And one way we thought of is, why not try reducing ZIP14 expression specifically in the liver? And the rationale is because hepatocyte-specific ZIP14 knockout mice don't load manganese in the brain. This has been published. They don't load it because if you block hepatobiliary excretion of manganese, there are alternative ways that manganese can be excreted. And one way is via the intestine. And that is also mediated by ZIP14, but that's a totally different issue. The other reason why we thought, let's just knock it down in the liver, is because the liver is not only the main site of iron loading, but also the primary site. So this is the first organ which gets the chance at taking up iron. If we inhibit it there and then chelate, it's possible that reducing ZIP14 expression only in the liver, which will increase plasma NTBI levels, will be sufficient to increase the chelation and excretion of excess iron. That's at least the idea. And to test, we can use either hepatocyte-specific ZIP14 knockout mice, or in theory, we could use hepatocyte-specific ASOs, such as GalNAc conjugated ASOs, plus or minus DFP treatment. We thought the simplest route was let's just use the genetic model and be done with it. And so this is the study design. We took male mice. These are at 4 weeks of age, 6 to 7 mice per group, and we allocated them to these groups. So basically we had control mice, hemojuvelin knockout mice, and we crossed hemojuvelin knockout mice with the hepatocyte-specific ZIP14, and also took these two groups and coupled them with deferoprone to see what would happen. And DFP was administered as before in the drinking water, and then we measured tissue non-heme iron levels. And in this study, it's a little different. We added some more tissues, thinking that, oh, maybe iron is going into the muscle or the lung, and we're not measuring it before. And in this study, we also measured total carcass iron. I can tell you about how I did that later if you'd like to know, because it's a little different than most people do it. But okay, let me just show you the data. Bottom line is that if you have the hepatocyte-specific knockout of ZIP14 in the context of hemojuvelin knockout, you decrease iron loading in the liver. This is what you would expect. If you treat a hemojuvelin mouse with deferoprone alone, iron decreases. What happens in other tissues? Well, pancreas, you really don't see an increase in pancreas in this model compared to the hemojuvelin knockout. You add deferoprone, it goes down. You have the double, it goes down. So it's similar to here. What's interesting is that the spleen, for whatever reason, accumulated iron in the double knockout here. And also with the deferoprone, it really had no effect. The kidney was kind of the opposite. I know I'm throwing a lot of data at you, and the heart, I think the essential thing is that the liver-specific knockout plus DFP decreased it alone. DFP decreased it somewhat, but this was a little better. And we saw that in tissue. So, and here's muscle. We added muscle too. No significant difference. Okay, so I just threw a lot of data at you. What does it all mean? The bottom line is that yes, using the antisense or the liver-specific knockout, it prevents iron from loading in the liver. And more specifically, if you couple it with deferoprone, you still see a decrease in these extrahepatic tissues. What happens in the carcass? Are you just redistributing the iron? Well, here are the iron concentrations, the dried carcass, basically not much difference. You really don't see a huge increase in carcass iron concentrations when you knock it out specifically in the liver with or without deferoprone. You do see somewhat of an increase. What happens if you measure total organ iron? So now we have more tissues to add to our total organ. This is non-heme iron. Hemojuvelin knockout obviously have a lot. But what's interesting is if you just knock out ZIP14 in the liver, you see a great decrease in all these if you combine them up, more so than deferoprone treatment alone. And if you combine the hepatocyte-specific knockout with deferoprone, it decreases it a little more. But in this model, we also measured, okay, so we have a lot less iron in these mice, suggesting, as Caroline intimated, are they excreting more iron or possibly is it going into the carcass? And so in this study, we took the whole carcass, dried it down, measured total non-heme iron, and if you calculate the total in the carcass, we do see that they do increase the amount of iron, let's say about 300 micrograms, but it doesn't account for the over 1,000 that is less in these tissues, suggesting that we do have an increase in iron excretion somewhere. This is total non-heme iron. We also analyzed the samples by ICP mass spec or sent them out for analysis, and we see similar results. So the million-dollar question in this study is, can we prevent manganese from loading in the brain? And the answer here are the manganese levels. Actually, that's, yeah, that is the brain. These are brain manganese levels. And you can see that deleting or inactivating ZIP14 in the liver does not increase brain manganese levels. And if I'll remind you of the data from the ASO study, ASO1 did increase brain manganese levels and plus DFP even more. So this, although it decreases total body iron levels, it seems it doesn't have this adverse effect of altering brain manganese levels. So overall conclusions, this is the last slide, is that 4 bullet points: treatment of iron-loaded hemojuvelin knockout mice with ZIP14 ASOs enhances DFP-mediated removal of tissue iron in tissues that are affected by iron-loaded pathology, the liver and the heart. And so these data provide in vivo non-clinical proof of principle for benefit of using combined or ZIP14 ASOs as an adjunct to standard chelation therapy in the treatment of iron overload. The ZIP14 ASO-induced alterations in manganese homeostasis may be avoided by using liver-specific ASOs. This is something we can test, or possibly by fine-tuning the ASO dosage or design. There are other ways to approach this, but first, that's what we showed. And lastly, a reduction in ZIP14 levels alone appears to decrease total body iron in hemojuvelin knockout mice, and this is probably by an increase in iron excretion, which we still need to measure formally. I'd like to acknowledge all these people. I'm not going to read off everybody's names, But I would like to highlight Qingli. Qingli did the lion's share. Essentially, all the work that I showed you today was done by my fantastic PhD student who I wouldn't let leave, so she's now my postdoc. And thank April Pharma and Michael Spino for providing the Ferroprone, and the folks at Ionis for kindly working with me on this project. And last but not least, of course, NIDDK for funding my research. So thank you.
Thanks, Mitch. Great talk. Really, really thought-provoking, actually. I have to think about it a bunch. We have a couple of questions in the chat that I want to invite people to ask. Tom, do you want to ask yours first and then Ella after that?
Is that Tom Gans or Tom Bartnikas?
Oh, it's Tom Bartnikas.
Okay.
Sorry, Tom.
No problem. Whenever I hear people talk about Tom, I assume they're talking about Tom Gans. So Mitch, I have a sort of a broader view question. What if in conditions of iron overload, the liver is really the best place for excess iron in terms of the overall health of the individual? And that if knocking down ZIP14 expression in the liver and or other tissues results in the redistribution of iron to places where it's even more toxic. Do you have thoughts on that?
Oh yeah. We pointed that out in the last paragraph of our cell metabolism paper, that you can't, you can knock down ZIP14, but it has to be used in combination with an iron chelator because of this redistribution effect. And I think what our data show is that if you do combine them, you mitigate the iron redistribution to other tissues. And in fact, what it seems is you accelerate its body iron losses. So yeah, I mean, that was the initial concern all along. You can't just knock down ZIP14 because it's just gonna go into other tissues, most notably the heart. That's a bad thing. But if you combine it with the iron chelator, it seems to be okay.
Yeah, and another quick question. Actually, pretty big question. Have you specifically looked at rates of iron absorption and excretion?
No.
Yeah.
We're going to do that.
That'll answer your question.
Well, yeah, that's what we're going to do once we can get some now super costly iron 59.
Thanks, Mitch.
Okay, Ella.
Hey, Mitch. Have you measured hepcidin in these mice where you have redistribution of iron to the spleen? And why would hepcidin be more effective if it's increased? Why would it be more effective if hemojuvelin is knocked out there?
Oh, yeah, well, okay. We did measure hepcidin in all of our models, either the total knockout or the hepatocyte specific knockout, and it's similar to the wild type controls, indicating that hepcidin induction is independent of iron loading in hepatocytes. But to answer your question about the spleen, why iron is loading in the spleen, and I'll refresh your.
So what you're saying is hepcidin is higher than in.
It's these data right here. Oh no, it's not. It's not these data. I'm sorry. It's the hepatocyte specific knockout, which is here. Just so we're all on the same page. Yeah, right here. Mm-hmm. So yeah, so why, first of all, why is iron loading in the spleen when they, hemojuvelin knockout, they wouldn't make hepcidin efficiently. Why is it loading when you have so much ferroportin to pump it out, right?
But is hepcidin, well, my question is also, is hepcidin higher there by any chance?
And why
not. Would it? Okay.
It's not.
So it's hepcidin independent spleen.
It's hepcidin independent. I'm pretty sure about that. I guess if Qingli's here, maybe she can correct me, but I think it's not different. We thought of that. That's are you saying splenic hepcidin mRNA levels?
No. I meant systemic hepcidin.
Okay,
Whether
yeah.
I was
Well, we
wondering whether
haven't-
you overcame hemojuvelin
Yeah.
knockout to produce more hepcidin
Yeah,
in some way.
yeah, we can't explain that. It's kind of weird, but
Okay.
good question.
A second question, you went over those other metals in hemojuvelin knockouts faster. I was just wondering, in models where you have a lot of NTBI and now ZIP14 is participating in transport of iron, is metal homeostasis of those other metals like manganese affected at all, just
Yes.
by sheer presence of NTBI?
Yes. Yeah, if we have ZIP14 knockout, iron-loaded mice, either by iron loading by diet or by hemojuvelin knockout, and in those models you have excess plasma NTBI, but on the other hand, since you don't have ZIP14, you have excess manganese as well. If you look at brain metal concentrations, what you do see is more iron, less manganese. Translation, they're competing for metal uptake into the brain via a different transporter.
All right. Thank you.
Other questions? Anybody feel free to unmute yourself at this time?
Hi, Mitch, this is Jody. Great talk. I
Hey,
really
Jody.
enjoyed it. I have a question that's kind of unrelated to the subject matter of your talk, but just about NTBI transport in general. Do we know anything about how NTBI is taken up into the kidney? What transporters are relevant or what you might be thinking of? Is this something where it comes up from the urinary surface? Is it coming from the blood? Do we see changes in urinary excretion of iron in these models?
Yeah, that's a great question. It's something that we have been tangentially studying over the years. ZIP8 appears to play a role. I mean, we have ZIP8 knockout mice crossed to hemojuvelin knockout mice, and they load less iron in the proximal straight tubules in the kidney. But overall, they load similar amounts. So it seems to be only in proximal straight tubules. So ZIP8 appears to play a role. ZIP14 may also play a role too. Maybe kind of the data that I showed today is that we're having an increase in iron excretion in at least the total body knockout. Maybe that's facilitated by loss of ZIP14. And you know, we in the lab we call manganese. Manganese is kind of our substitute for NTBI because manganese and NTBI are transported very similarly. And what we did do is we looked at urinary excretion of manganese in ZIP14 knockout mice, and it's increased. So if manganese is like NTBI, we would expect NTBI to also be increased, although we haven't measured it formally. I don't think we have. Maybe we did.
Are these transporters, do you think they're mainly happening on the urinary surface where it's reuptake of iron, or do you think this is happening from the blood? Or do we have any idea about that?
I think it's a reuptake from the urine or pro-urine. So after being filtered, it's taken up either by ZIP8 or ZIP14. I can tell you that ZIP8 does localize to that surface. So it likely plays a role in proximal straight tubules, and it's induced by iron. Still haven't published that, but that seems to be the case. Yeah, we're still trying to figure that out.
Thanks, Mitch.
Mitch, this is Tom Gans. How about the role of ZIP14 in the embryo and the placenta?
Yeah, you know, in the placenta, I don't know about the placenta. It's something that we've talked about a lot, is, you know, just harvesting neonates and measuring total body iron, but we've that's on our list, but we've never done it. We need to do it. My suspicion is that Z14 is probably dispensable for iron transfer across the placenta. ZIP8 probably plays a role. Who knows what?
Okay, Mitch, do you want to stop sharing your screen?
Yes.
And everybody can kind of say hi and ask any other questions they want. Anybody else have a question? Okay, I want to thank you all for your participation, and I posted in the chat and want to mention here, for those of you who are trainees and are interested, there's a new opportunity for a postdoctoral fellowship at Carolyn Philpott's lab. She's waving right there. So if you have any questions, reach out to her. I will also get it posted up on our website so that you can go read about that. There are other individuals on the screen I see here who also have postdoctoral positions open. So I highly recommend you check out our websites to see if there's any opportunities for those trainees who might be interested in staying in the iron hematology research field. And it's so nice to see everyone. Want to wish you all good luck. Unmute yourselves, say hi.
Can I mention one more thing? There will be East-West Iron Club, EWIC, at the beginning of October, and it will be virtual. It will be great to see you all. So hopefully you'll come.
Yeah, and that's also on the website. So we've put it up there for everybody to take a look at in case you need another link to it, because those are great meetings, wonderful opportunities to connect.
And in case you missed it when I mentioned it earlier, is that there's also the Gordon Research Conference. Supposedly, it's still going on. It may change as of this afternoon. I'll know, but it's a go so far. There are, there's iron will be there, manganese and other metals. But if it does go, please consider attending. I think the deadline is in 5 days if you'd like to register.
Mitch, if
Mitch, I gotta say thank you very much for what is probably going to be the biggest headache in organizing a meeting, that you ever had to do. I know you didn't sign on for this, but we really do appreciate you and Karen working your way through this when it's been so difficult. Thanks.
Well, thank you. It's mostly been Karen. Karen's been fantastic. But yeah, it's been kind of a headache juggling everything, and the juggling's still not done. So we'll know more about it today.
Mitch, if you learned something new that you would like me to send out on social media from the CIHD regarding the Gordon Conference, just send me an email and I'm happy to get it posted up on that site too, to inform.
Will do. Thank you. Thank you.
Sure. Okay, everybody, nice to see you all again. Hopefully some people will get to see each other in person soon, and virtual hugs to everyone. Take care.
All right. Thanks, everybody.
Thanks.
Bye.
Bye. Appreciate it. Bye-bye.